Hello everyone, Dr. Alex Vasquez here with the second video in our program on human microbiome and dysbiosis in clinical disease. In this second video, I'm going to focus on physiologic and pathophysiologic responses, so I'll begin with the usual necessary repetitions and then we'll dive right into the coursework with at least one contextualizing case. As you all know, this information is protected by copyright and published in my books. Some of it's also protected by trademark, and the scope of this information is intended for licensed clinicians, health science students. This is not individualized health advice, and doses are for adults unless specified otherwise. I've worked as a researcher and consultant lecturer for Biotics Research Corporation. Some of the treatments that I discuss might be considered off-label or unapproved by the US FDA, so prescribing physicians will have the ultimate responsibility to determine the appropriateness of drug prescribing per patient. Recall that many of the more intricate and intimate details are covered in the printed clinical monograph, which also provides you access to more than 12 hours of password-protected videos for additional explanations and clinical application. As we're going through the material, you can always pause or replay the video to let the information sink in at a deeper level, or if you want to take the time to read each slide word for word. As I mentioned before, what I'm aiming to provide you here is a structured approach to dysbiosis and the human microbiome and ways to integrate this information with a more comprehensive treatment protocol, which I've discussed and defined as the functional inflammology protocol. As I mentioned before, and as we all know, a lot of times in our clinical practices, we're dealing with disorders of sustained inflammatory responses, whether those are metabolic, allergic, or autoimmune. And so the pharmacocentric model of dealing with those conditions is basically to block the reception and release of those inflammatory mediators. I call that trying to intercept the inflammatory football. So we can spend time doing that, and that's certainly one way to practice, and at times that's a necessary way to practice in times when patients are having acute inflammatory or allergic responses. But we certainly don't want to practice like that forever, because if that's all we ever do, then that's all we'll ever accomplish is crisis management. And noble as that is, we need to actually try to get these patients out of their inflammatory situations, and in order to do that, we have to look upstream at these primary causes of immune activation. And of course, a lot of these can work in concert with each other to promote sustained inflammatory responses, previously termed chronic inflammatory diseases. So again, of course, what we're focusing on here is the dysbiotic component of sustained inflammatory responses. In order to help all of us memorize this information and apply it clinically, I've developed this functional inflammology FIND-SEX acronym, which of course you can also remember as FIND-SET, as if you're searching for a set of problems, FIND-SEX or FIND-SET. Of course, that stands for food, infection, nutritional immunomodulation, dysmetabolism, and dysfunctional mitochondria. The S category stands for lots of different things like style of living or lifestyle, sociology, sweat as a metaphor for exercise, stress modulation and sleep optimization, and of course, specialized supplementation and also surgery at times. The E stands for endocrine, and the X can stand for xenobiotics, or you can substitute the X for a T and have that stand for toxins if you choose the FIND-SET acronym. I'm sure all of us are familiar by now with the SOAP note that we all learn in school, subjective objective assessment and plan. This is a way to organize and structure our patient intake and ultimately the assessment and plan as well. So what the functional inflammology protocol provides is a structured approach to the plan component of our patient management, because of course we all know that we can use pharmaceutical medications, we can follow up with tests, referral, etc. But what we need is structure in order to recall that we need to focus also on food, these persistent infections and dysbiosis, nutritional immunomodulation, which is relevant for allergy, asthma, and metabolic inflammation, such as we see in insulin resistance and diabetes. Also, dysfunctional mitochondria and dysmetabolism, stress, psychosociology and style of living, and also sleep optimization, endocrine and hormonal imbalances, and the total toxic load and total xenobiotic load. My definition of dysbiosis is that it is a relationship of non-acute, non-infectious host microorganism interaction that adversely affects the human host. We could abbreviate that slightly by saying that dysbiosis is a relationship of adversarial, we might say, host microorganism interaction, or we could say that dysbiosis is a relationship of host microorganism interaction, again, that adversely affects the human host. So the concept of dysbiosis, in my opinion, can't be explained simply by looking at bacteria and other microorganisms. We have to look at the effect that those have on the human host. And that helps us have a more broad definition of dysbiosis, and that gives us more opportunities to intervene clinically. We want to understand the role that the total microbial load plays in the genesis and perpetuation of sustained inflammatory responses, and we want to be able to define dysbiosis beyond its microbial definition. And when we do, we'll see that we have other treatment options available to us other than, number one, focusing on the microbes. Number two, we can restore immune function so that the immune system does its job of controlling and killing off these adversarial microbial populations. And number three is we can promote immune tolerance to these otherwise benign microbes that often serve as triggers for sustained inflammatory responses. In 2006, I published a book and an article discussing what I call multifocal polydysbiosis, and that's because we now realize that dysbiosis can occur in numerous locations via numerous different microbes and can also occur via different molecular mechanisms, and I'll discuss some of the most important of those in this presentation. The total microbial load from all of these different locations gets absorbed by the body and has to be dealt with by the immune system. So when the immune system and the body as a whole has to deal with all of these major microbial molecules, eventually we move from physiologic responses into pathophysiologic responses, especially when the total microbial load is very high or when skin and mucosal barriers are breached so that more microbial debris is absorbed. And thirdly, also, when the patient is in a pro-inflammatory state and their immune system is less tolerant of these microbial molecules, we're more likely again to see the transition from physiologic responses into the pathophysiologic responses that lead to clinical disease. Based on the patient's physiology and genomic pattern, along with their microbial pattern and the extent of that microbial pattern, we see these conditions basically filter into several different categories, which I've defined here as clinical prototypes, including encephalopathic prototypes, central sensitization or pain prototypes, also inflammatory immune complex disorders, and the classic prototype, which is reactive arthritis and its autoimmune variants, such as ankylosing spondylitis and rheumatoid arthritis. As I've said before, often what we find when working with autoimmune and inflammatory patients is that they are having pathogenic inflammatory responses to non-pathogenic microbes. We see this in a variety of inflammatory and autoimmune conditions, and again, we want to extend our definition of dysbiosis-triggered disorders to look beyond the microbe. So first of all, we start with considering the microbe. We can kill off the bad ones, we can add more of the good ones. Number two, we want to support immune function so that the immune system does its job of controlling and or killing off these quote-unquote offensive microbial populations. And number three, we want to promote tolerance to these benign bacteria, and that's the immunotolerogenic protocol or nutritional immunomodulation. And with that, I've reached the end of our quick review, and we're going to dive into the new material that we have here on pathophysiologic responses. So one of the ways to understand these pathophysiologic responses, and in my opinion, this is the easiest way to understand and the easiest way to explain to patients, is to use the NF-kappa B pathway as a model. And you can see that outlined here. I've published this in my books. You have it in the printed clinical monograph. I also have an article that I published back in 2005, which is available online if you want to take a look at that. But let's look at NF-kappa B as a prototypic transition, let's say, from microbial molecules ultimately into these clinical events that you see summarized and exemplified at the far right. So, a variety of environmental and microbial triggers can coalesce or work independently to activate this NF-kappa B pathway. NF-kappa B exists within the cytoplasm of the cell. When it gets activated, as we say, it translocates into the nucleus and binds directly onto the DNA to increase the expression of pro-inflammatory genes. So NF-kappa B stands for nuclear transcription factor kappa beta. And again, this binds directly onto the DNA to trigger the increased expression of certain genetic patterns, we might say, and in this case, those patterns are related to inflammatory responses. Whether that's interleukin 6, cyclooxygenase, interleukin 1, lipoxygenase, INOS to increase production of nitric oxide, or tumor necrosis factor alpha. These are the inflammatory mediators that ultimately lead to these clinical disorders that we deal with in our practices. So, as I'm sure you all know, we have lots of drugs that we can use to manage these clinical events, and sometimes those are necessary. We have other anti-inflammatory drugs that we can apply here, but again, in the naturopathic and functional medicine model, we try to look upstream and inhibit or reduce the activation of NF-kappa B de novo, as we might say. If we never address the upstream triggers of inflammation, then we're going to spend our entire professional lives trying to intercept these inflammatory footballs and do damage control with these clinical outcomes, whereas we could focus at least part, if not most of our attention at the upstream triggers of this inflammatory response. This inflammatory response is not a mistake. It is correct. So basically what we do when we're using anti-inflammatory drugs is we're trying to kind of impose order on a system that's actually already ordered. It's already functioning correctly. We're trying to block the expression of an orderly system by using drugs that block inflammation here, whereas we should be addressing the causes of this response, not trying to interfere with a correct response. So, as I'm sure you all know, we have plenty of drugs that we can use here to manage these clinical events. We also have an entire class of anti-inflammatory drugs that we can use here. But as I said before, in the naturopathic and functional medicine model, we try to look upstream at the actual triggers of this very appropriate inflammatory response. So, one of the ways we would describe this in naturopathic medicine is to say that this response is not a mistake. And these clinical outcomes are the logical extension of an appropriate inflammatory response to an excess of triggers. So we should be addressing these triggers rather than trying to suppress a physiologically normal response that becomes pathophysiologic when in excess and in a pro-inflammatory state. And of course, a lot of our patients basically live a pro-inflammatory lifestyle, and I've discussed that in the books, and of course, it's been published in other articles by other authors as well. Before we leave this page, in order to summarize what I again consider to be a very simple model, we look at all of these triggers activating certain inflammatory responses, changing gene expression, leading to the elaboration of excess inflammatory mediators, and of course those mediators have physiologic and pathophysiologic consequences. When in excess, these inflammatory mediators trigger clinical disease, and that's of course what we deal with. So the question becomes, where and when do we want to intervene? Do we want to intervene at the last moment and really just treat disease for the rest of our professional lives? Do we want to intervene here at the level of inflammation? Or do we want to look upstream at the actual triggers of this either appropriate or excessive inflammatory response? And when we do this, when we actually look upstream, only then do we provide our patients and ourselves an opportunity to actually cure these diseases rather than quote unquote medically manage them for the rest of our lives and for the rest of our patients' lives. Several of these inflammatory pathways are defined in more detail within the book. In order to give some structure and some clarity to the way that these microbial molecules trigger human responses, let's look at the example of toll-like receptors. These are transmembrane proteins that sense occurrences within the extracellular environment and translate those into intracellular responses, one of which is the activation of NF-kappa B, which we looked at on the previous page. So, for example, when bacterial endotoxin activates toll-like receptor 4, that triggers a cascade of events, ultimately leading to activation of NF-kappa B, NF-kappa B then translocates from the cytoplasm into the nucleus, changes gene expression by direct binding onto DNA, and from there we see the elaboration of an excessive or increased level of inflammatory mediators, and those inflammatory mediators then contribute to clinical disease. So what we can do with this clinically, in addition to looking for the triggers of this inflammatory response, is we can actually interfere at this level, for example, by using fatty acids and certain nutrients to downregulate the excessive response of these toll-like receptors in their activation of NF-kappa B. We can also use a variety of nutrients to directly inhibit or modulate the NF-kappa B response. And of course, then we can always continue to go downstream and use certain nutrients, botanicals, or pharmaceutical drugs to block the reception and release of those inflammatory mediators. But what I think is clear is that we actually provide better medicine when we look farther upstream. Rather than keep our patients dependent on anti-inflammatory treatments, let's look upstream at the actual causes of that inflammatory response and give our patients the opportunity to actually become liberated and free of their disease, rather than simply to have their diseases managed for the rest of their lives. Let's look at molecular mimicry and cross-reactivity as an exemplary model for the induction of pathophysiologic responses by microbes. So basically, the take-home message here is this: that microbes produce in their proteins and in their cell membranes, for example, certain amino acid sequences which are identical or very similar to the amino acid sequences found in human proteins. So when the immune system begins to attack or have an immune response against those microbial molecules, some of that immune response, we might say, either misses the target or spills over to affect the human tissues that share those same amino acid sequences. So, amino acid similarity or amino acid sequence homology is the physiologic and molecular basis for molecular mimicry, which is then the basis of immune cross-reactivity. So, as I state here, similar human and microbe amino acid sequences induce immune cross-reactivity when the immune system targets the microbial sequence and then hits the human tissue antigen instead. And you can see that beautifully exemplified in this photo. So, we could say here that the immune system is trying to hit this target, but some of these antibodies and the antigen-specific immune response actually ends up hitting the human instead. So you'll see lots of conversations in the literature that discuss specifically molecular mimicry and immune cross-reactivity. Again, when the body is attacking these microbes, some of that immune attack accidentally targets the identical amino acid sequence that happens to be found in the human host. And that's why we see certain genetic patterns or certain tissue markers in humans associated with autoimmune diseases induced by microbes. And the perfect example, the best example, is that of HLA-B27. HLA-B27 is a human tissue antigen which shares amino acid homology with microbes such as Klebsiella pneumoniae and E. coli. Again, before we leave this page, we want to appreciate that amino acid sequence homology or amino acid sequence similarity is the basis of molecular mimicry, and that is the basis of immune cross-reactivity. Let's look now at a case from my own practice that exemplifies this very well. This is a 40-year-old man with idiopathic peripheral neuropathy. This is a 40-year-old white male who presents with a 5-year history of idiopathic peripheral neuropathy, manifesting with numbness, weakness, and tingling. So you can see immediately that this is a mixed motor and sensory neuropathy. He notes that this is exacerbated by accidental consumption of shellfish and associated with fever. Now, in my opinion, this short history alone gives you a model that you can use to understand and address this case. I recall very clearly when I was working with this patient that immediately upon receipt of this history, I knew exactly what was going on with him. And then later, of course, we confirmed that with laboratory testing and empiric treatment, which turned out to be successful. And therefore, empiric treatment became not simply therapeutic treatment, but also a diagnostic trial to confirm my assessment. My presumption was that this patient had a true food allergy to shellfish, and that when he consumed shellfish, he acquired a allergy-driven leaky gut, and that leaky gut promoted the absorption of microbial molecules from his gut, and he was having an immune cross-reactive response to those microbial molecules, and that's what was causing his idiopathic peripheral neuropathy. This patient had some slight evidence of autoimmunity. I believe he had a slightly elevated anti-nuclear antibody, maybe a ratio of 1 to 80, something very low. And I believe his brother also had some type of inflammatory disorder as well. By the time he came to see me, he had already spent at least $10,000 on neurological consultations, MRI, CT scans, lab tests, and lumbar puncture. Our goal, of course, was to cure him and provide some preventive health care as well. And as I said before, I think all the information provided here in point number 1, which I received within about the first 5 minutes, I think that alone is sufficient to provide a diagnosis and at least a provisional means of assessing this patient and treating this patient, which we could, of course, implement basically on the first day. Now let's take a look at some of the lab tests and evaluations. We performed a comprehensive cardiovascular profile and found that he had slightly elevated fibrinogen and high sensitivity C-reactive protein. That's exactly what we would expect for a condition of sustained low-grade inflammation. I asked this patient to do a stool test because I was already convinced that he quite likely had gastrointestinal dysbiosis. He refused to do stool testing, but he was willing to do a lactulose mannitol assay, which we performed. Anytime we think about leaky gut, we want to think about the causes, the most common causes of leaky gut. Those include inflammatory bowel disease and celiac disease, overconsumption of enterotoxins such as NSAIDs and alcohol, clinical malnutrition, acute illness, trauma, and sepsis, also food allergy, again including celiac disease, and any type of quote-unquote parasites or dysbiosis. You'll notice that many of these can be excluded by a good history and physical exam, so in my opinion, that makes us focus on food allergy and parasites. I was already pretty convinced that this patient had food allergy, and so I wanted to understand better whether this patient did or didn't have gastrointestinal dysbiosis. I was pretty convinced that he did have dysbiosis. Again, my model for assessing this patient is that he had food allergies, and when he consumed foods that he's allergic to, in this case specifically shellfish, that triggered a leaky gut, and that triggered the absorption of more microbial molecules, and he cross-reacted with those microbial molecules to produce this idiopathic peripheral neuropathy. So here you can see the results of his lactulose and mannitol assay. You can see that his lactulose percent recovery is quite high, so that correlates with a leaky gut. Remember, I told you before that lactulose rhymes with leaky. And so this indicates mucosal damage and increased permeability or mucosal hyperpermeability. You also see that his mannitol percent recovery is on the low end, and I mentioned to you before that mannitol rhymes with malabsorption and malnutrition. So when we see this pattern of elevated lactulose recovery and reduced mannitol recovery, that's consistent with mucosal damage and villus atrophy. What this provided me was clear evidence that this patient has something going on in his gut, whether it's an allergic response or an inflammatory response. And with this data, he was then willing to perform a comprehensive digestive stool analysis with comprehensive parasitology. And with that, we were able to move to the next stage of managing this condition. Very importantly, what this provided us, even though I was already convinced just based on the history that this patient had leaky gut, is that it provided the patient with proof that we needed to go ahead and do a comprehensive digestive stool analysis with parasitology in order to look for the cause of this mucosal damage. So at this point, we had specific evidence of low-grade inflammation with the elevated fibrinogen and high-sensitivity C-reactive protein, and now we also could pinpoint the location of the damage to his gut. We had evidence now of leaky gut or intestinal hyperpermeability. And again, we were then able to move forward and do comprehensive stool analysis, and our findings there were very significant. He had elevated secretory IgA. He also had elevated lysozyme, indicating an inflammatory response within the gut. So he has an immune response within his gut with the elevated secretory IgA. He has generalized inflammation within his gut with the elevated lysozyme. And then the question becomes, can we find a microbial trigger? And in his case, he had Pseudomonas. As you can see here with his stool test results, let's start with the beneficial bacteria on the left. He's got good probiotics, good bifidobacteria, reasonably good lactobacillus. In the middle column, I don't see any major problems there, but the Pseudomonas is definitely a problem, and he's at +4. We can also look at the bottom left, and we see that he's got some abnormal yeast, Candida glabrata and Rhodotorula. I consider those just indicative of a generalized mucosal or systemic immune suppression. Those are not normal yeast. We typically don't see those. And so when we do see them, it's indicative of some type of immune impairment. But again, my biggest concern here is with the Pseudomonas, especially in this patient's case of this so-called idiopathic what appears to be an immune-mediated peripheral neuropathy. So the molecular connection between Pseudomonas and peripheral nerves is that the Pseudomonas bacteria shares amino acid sequences with myelin basic protein, as you can see evidenced here in this article discussing multiple sclerosis. So Pseudomonas produces an enzyme called decarboxylase, which again shares amino acid homology with myelin basic protein, which of course is a protein found in peripheral nerves. So I think what we can reasonably state here is that this patient might be having an immune cross-reactivity response based on amino acid homology between this enzyme produced by Pseudomonas aeruginosa and the amino acid sequence found in myelin basic protein. So as you can see here, this article was published in November of 2001, Clinical and Diagnostic Laboratory Immunology. Let's look at another article published in Journal of Neuroimmunology in 2003, Cross-Reactivity Between Related Sequences Found in Pseudomonas aeruginosa and Myelin Basic Protein. Again, let's look at some of the details here. We see that Pseudomonas aeruginosa can cross-react with Myelin Basic Protein based on a sequence found in an enzyme that it produces, and that's a decarboxylase enzyme. So in other words, Pseudomonas produces this decarboxylase enzyme,